At this time, we will reverse the order since Dr. Fagan went first and Dr. Afzali. We'll let Dr. Afzali go first on any final rebuttal she has to say that she won this argument with Dr. Fagan, and then we'll let him close and we'll go to audience questions.
Vote for me. Is this on? Is this on? Oh,
I think so.
Okay,
Yeah.
still, vote for me.
Vote for her, I don't care.
You're both killing me.
I think one of the issues that really we hopefully both would agree on is that I think the whole framework for the biosimilar approval has kind of gone off the rails. And, you know, I think it's interesting forensically to look at the origin of this. And when I see, it's the same folks that developed the approval process for generic drugs that wrote the book on biosimilar approval. And you can see terms philosophically creeping into it. There's a concept called assay sensitivity, and Anita alluded to that. And she made, I think, a very good case that rheumatoid arthritis is not the most sensitive assay of population, in that ulcerative colitis probably is the assay sensitive to stress tests of biosimilar because of the pharmacokinetic problem that she alluded to. But, you know, the fact that they use a term like assay sensitivity, well, that's coming right out of generic pharmacokinetic approval. And then to insist on equivalency studies, which is, you know, by nature are two-sided, they're very statistically inefficient. When all the clinician wants to know is non-inferiority is really kind of silly too. But it comes from two-sided PK testing of small molecules because you want to know that the generic drug doesn't produce too high concentration or too low a concentration. So you have to have Goldilocks in the middle. So, you know, I think that's where I kind of object to the whole process of where we are. And if we had to do it over again, I would have been a lot happier to use endoscopy in ulcerative colitis. But the reality is we are what we are. We're there now, so deal with it.
So, audience question. I have a few on therapeutic drug monitoring, and I think the general sense of the questions are, we're expecting multiple biosimilars, and we have therapeutic drug monitoring assays already. Would you use the drug assays that are available for the innovator, and do you expect it to be accurate?
I think that's our only option at present. And so, yes, we will continue, and we should continue to use the assays. What I wanted to highlight was basically just that there still remains variability, and regardless of whether it's with your biological product, the innovator, or your biosimilar, you should still be able to and should continue to perform therapeutic drug monitoring, look for good detectable levels, as well as evaluate for antibody development.
I think you're okay as far as the drug concentrations, because these are all TNF capture assays. So they bind TNF, and if you bind TNF, it'll be fine to detect drug concentrations. Where it all falls down, though, is the immunogenicity. I have no idea what they're measuring. And we know that from Planetra, in that the rates of immunogenicity were far higher than anything we've seen before with infliximab.
So when you say you're not sure what to do with the immunogenicity, so let's say you have biosimilar, whatever it is, infliximab, adalimumab, and you send a therapeutic drug monitoring test with what we have now, and you get low-level antibodies, moderate antibodies, high-level, what?
I just don't know how to interpret the antibodies. I mean, if it's clear-cut, if you've got very high levels of antibodies and no drug, but then, you know, anything intermediate, I don't think we understand.
Okay. So another question on, the presumption is there will be multiple biosimilars within each group, right? So infliximab, adalimumab. And the question is, so you guys have shown us you can do one switch between innovator to biosimilar. What about when you have 3 or 4 of them and you're going innovator to biosimilar A to biosimilar B to biosimilar C? Is that a concern?
That is the concern.
That is the concern, right.
Okay, and so the second corollary to that is, if you have a patient that you're being told they're on the innovator drug in 2017, and the question, somebody says you have to use biosimilar. What is your response to that?
Well, I had not been in that situation yet, but I will be soon. And I'm going to put a note in the chart and have the patient sign it, and that we don't want to do this, and any consequences are therefore upon the payer.
So you would tell patient, I don't think this is a good idea.
Correct.
Dr. Afzali?
For a patient who's being asked to switch?
Yes.
Yes, absolutely. I don't think that we have enough data and enough validated good studies to demonstrate that switching is safe or efficacious for our patients.
Yeah. And I think it's a key point that the United States and Canada are not Europe. You're not going to see enforced, I don't think you're going to see enforced non-medical switching because of the liability issues.
It's very different. Yeah.
Question on specifically about the Switch aggregate studies, and I don't know if we can pull up either Dr. Afzali's or Fagan's slide on the Switch. This is specific to that. The question is, do I understand that it was not inferior, meaning the biosimilar, and that there were not more failures if you switched? Is that your interpretation of the Switch?
Yeah, to try to avoid the double negative. The study met its primary endpoint, and the hypothesis was that there was no clinical difference between the two drugs.
So you would say the Switch
study was a
says,
positive study.
okay. Dr. Afzali?
But as we pointed out, it was not powered to evaluate for the subgroups, and I know it's the S word he doesn't like, but it was not powered for that.
I'll repeat. That's really irrelevant.
So I can't remember which one of you alluded to there's drift already in the drug. So the question is there really a difference with the innovator drug to what we used, I guess, 10 years ago, 5 years ago, and now we go to biosimilars? So the question is, there's already drift in the innovator drug. Haven't we already seen the switching back and forth because we keep moving forward with a drift in the innovator drug for patients that have been on it?
Yeah. So the companies that manufacture these drugs would say we have quality control processes and we ensure that our product falls within these ranges. And for the regard to the glycosylation, basically they chop up the antibody and then they measure using high pressure liquid chromatography or mass spec the composition of the carbohydrates. And again, that's really not what we're interested in. Whether the soup looks the same, in 1999 versus 2009. What we're interested in is the pattern of the branching, because that's what the T cell sees when it decides to sensitize or tolerize. And there's no assay for that. So, again, it comes back to the snowflake. I think if I had to bet right now, I think the experiment's already been done. And that's why we have immunogenicity.
Because of the drift?
Because there's such heterogeneity already in the composition of the product.
Okay. So this is specific to Dr. Fagan. Why did Canada accept extrapolation of biosimilars in IBD after initially not allowing it?
Yeah. Well,
and what did you have to do with that?
That's a really complicated political question. Well, I'll be blunt. I think Canada took a very hard line on biosimilars. It was very conservative, was well ahead of the field as far as what the requirements were, and made it very difficult for biosimilars if you followed their original white paper guidance. When the EMA, really the house fell down, they approved across everything, Canada, the Health Canada was under tremendous political pressure. Why aren't we doing what the Europeans are doing? So they had to sort of save face, and there were differences in antibody-dependent cell-mediated cytotoxicity in Crohn's disease versus other indications. So they said, well, we'll approve for the arthritic indications and for psoriasis, but we're not going to approve for IBD. And then when the U.S. FDA made their decision, then they reversed themselves. It was all politics.
Politics. Another question. So this goes back to, we're talking a lot about switching, but when you have a bio-naive patient, do either of you have a preference about biosimilar, innovator? So again, naive to the product, does it matter? Insurance says or payer says, we want innovator or we want biosimilar. Do you care?
I think you just highlighted just that insurance or payer says, in a sense where for a bio-naive patient, insurances will only pay and the payer will only pay for whatever they want to pay for. So as much as what we want to do, Brian and I want to do for our patients, we will still perhaps be mandated by cost issues in regards to what we would want to give versus what the payer and insurance will actually pay for. And in that situation, for a bio-naive patient, then is perhaps worth less of a battle or fight, especially if the payer will only pay for the biosimilar product and not the original or innovator.
So in fairness to the biosimilars, let me ask slightly different. So insurance or payer says, we want you to start on biosimilar infliximab. A year later, they say, oh, by the way, we want you to go back to the innovator. So now you're switching the other way. Is that acceptable or still the same milieu?
No, I think it's non-medical switching.
Yeah.
Okay. So you don't have a loyalty to a brand?
No, I think the potential risk issue is the same, and until we have a proper crossover studies, I'm not going to agree to do that.
Okay. So speaking of risk, question, has there been any litigation by a patient, either against insurance, the provider, or something because of a switch that didn't go right?
Not to my knowledge, but you'd have to look up classaction.com to really get that answer.
Okay. Are there any better crafted studies or that are in the works or that we don't know about looking at multiple changes between IBD patients, not just all comers?
I don't think so. I think the vision of the future, I mean, a few years ago, a lot of us felt that there were going to, Anita's chart with 13 versions of adalimumab and 9 versions of infliximab. And so you could imagine a future where it's going to be a race to the bottom with 15 players involved. I don't think that's the way it's going to shake out. I think the margins, if you put all the support services and everything in the price of the drugs to manufacture, there's a threshold level that you're not going to go below, and probably 13 companies are not going to survive. It's probably going to be 2 or 3 of them. And probably they will be the companies you know now. It'll be the big pharma players. And those folks have a vested interest in not causing problems. And, it's very expensive if you're Pfizer and you end up with a problem where you've sensitized the whole cadre of patients. So I think the innovator companies will actually drive the field, and they will probably do the study that Anita and I were advocating, because it's in their best interest to do so.
So, and this is probably a separate answer from each of you. So it's been reported that there's 15 to 25% less expensive for a biosimilar. Who actually saves the money? So we have U.S., we have Canada.
That question remains and to be determined. Even from the slide that I demonstrated to you, this doesn't take into account as far as the industry's rebates and whatnot that for the originator product that could still be offered. And the question remains whether that rebate would also be offered with the biosimilars for the patients. But I think it's to be determined in regards to where are the actual cost savings and who will actually reap the financial benefits.
In Canada?
Oh, I don't think that Canada's any different. I think that 2/3 of Canadians have private health insurance for drugs. So it's going to be the same issue. It's probably the payers are going to be the big winners.
Probably the payers, okay. So I think this is angling at where Dr. Afzali had mentioned, hey, in the subgroup analysis, maybe the biosimilar didn't look as good. So is there enough data to compare, to, I guess, estimate the numbers needed to treat when you switch that you're causing harm? Meaning, is it if you switch 10 patients, you've caused harm to 1, 5?
Yeah. Well, you can't calculate a number needed to harm unless you are certain that the effect is real. And I don't think there's any evidence that the effect is real. Anita kind of cherry-picked the data a little bit. When you actually look at when you actually look at the results of NorthSwitch, that is the only endpoint. Actually, if you looked at all the other endpoints, that actually that pedogram that she showed favored Crohn's disease. It actually, for all the other endpoints, it didn't do that. So I think really, it's probably random, not real.